No one is guaranteed an easy life, but we can make it easier for others. Many individuals from far-flung places suffer from infections. One of the causes of these infections are wounds. Access to the treatments, such as an antiseptic, is hard for these rural areas due to the geography and the economic status of the region. So, for the microbiology project too, we thought of developing something that is local within these areas, something that is cheap for these communities, and something that is sustainable and renewable. It is the development and research of a banana sap antiseptic. Growing need is further exacerbated by the reality that a country such as the Philippines, which is a third world country, does not have a proper response to this growing need for alternatives to antibiotics due to the antibiotic resistance that these hemolytic microorganisms that infect wounds have developed. In addition, one of the foundations of the project is the need to reach these far-flung regions.
Okay, the project aims at introducing this technique of wound treatment to them since it is available to them and is commonly found in rural environments. So it is also something that is sustainable since this natural material can be utilized throughout its whole life cycle. And in addition, the fruits of the plant are used for consumption and its stem parts are used for developing the antiseptic. And lastly, it is easily renewable because these plants grow back easily, and since the material, the sap, is a natural material, its source can be like replenished over and over again.
How was the project done? Four possible solutions of antiseptics were looked into carefully in the lab. These four solutions were solutions of sap with ethanol, sap with acetone, sap with 10% sodium chloride, and a raw sap solvent. There are two parts to see how effective the sap were in preventing the growth and spread of bacteria. In each of the experiments, there are two trials done to confirm the results of each. The solvents were tested against 5 bacteria and 1 fungi that are commonly found to cause wound infections, namely P. aeruginosa, S. aureus, S. pyogenes, K. pneumoniae, S. agalactiae, and C. albicans.
However, we wanted to narrow down on the most common ones that cause the most wound infections, and so we tested the efficacy of the solvents for against the Pseudomonas aeruginosa and Staphylococcus aureus species.
First, we tested if the solvents at different pH, specifically 4, 7, and 10, can inhibit the hemolytic activity of the bacteria in blood agar.
We wanted to see how active the sap's antimicrobial properties are in acidic solutions at pH 4, neutral pH, or the pH 7, whether the compounds can exhibit antimicrobial properties at pH 10. 20 microliters of each sap plus the solvent were added into the wells.
Final results showed that at pH 4, the solvents were most effective in inhibiting the hemolytic activity of the bacteria. All of the solutions with sap with ethanol, sap with acetone, sap with 10% sodium chloride, and a raw sap solvent were able to perform well in inhibiting the typical hemolytic activities of the microorganisms that we tested against.
So the typical beta hemolytic activity of these microorganisms were reduced to alpha and gamma hemolytic activity.
To add, at pH 4, all the solvents were effective, especially sap plus ethanol, in inhibiting the growth of the four microorganisms. pH 7, all the solvents were ineffective, specifically to Pseudomonas aeruginosa, Staphylococcus aureus, and Streptococcus pyogenes. However, all the solvents were able to inhibit the hemolytic activity of Klebsiella pneumoniae, Streptococcus agalactiae, and Candida albicans on pH 7 to almost no hemolytic activity.
At page 10, the solvents were ineffective, specifically to Pseudomonas aeruginosa, Staphylococcus aureus, and Streptococcus pyogenes species. But we were able to inhibit the rest of the microorganisms to no hemolytic activity. Next is we then further tested the solvents against the two most common microorganisms that cause infections in wounds, P. aeruginosa and S. aureus. This time we tested on ME-shaped plates in duplicates.
The results show that at pH 4, the growth of S. aureus was greatly inhibited, but not P. aeruginosa. The solvents were proven ineffective at pH 7, since P. aeruginosa and S. aureus species were able to grow in the media. At pH 10, solvents were able to inhibit the growth of S. aureus species, but not P. aeruginosa. We then performed a qualitative testing for phytochemical presence in the sap. We tested for 4 antimicrobial phytochemicals, namely tannins, flavonoids, cardiac glycoside, and saponins. We chose these 4 phytochemicals because they are known both in literature and Filipino ethnomedicine to stimulate skin growth and wound healing.
We tested their presence in different solvent mixtures to see what solvent is able to precipitate out more of these phytochemicals. So we performed the test by addition of ferric chloride for flavonoids, glacial acetic acid, and sulfuric acid for cardiac glycosides. And for saponins, it required like shaking of each solvent until there is formation of permanent foam. We were then able to obtain the following results. So the solvent that precipitated out more cardiac glycoside is the acetone solvent. Darker color indicates high presence of this phytochemical in the solvent. So for the tannins, 10% sodium chloride was able to precipitate out more of the tannins in the sap. Due to the darker color, all of the solvents were able to precipitate out of flavonoids in the sap sample.
Meaning, since we were able to test the presence of the cardiac glycoside in the sap, and since they are known to have been found to stimulate collagen synthesis and have the potential for wound healing, this suggests that acetone plus sap, when researched more thoroughly, has the capacity to both prevent bacterial wound infection and also stimulate skin regeneration.
In addition, we also performed a phytochemical presence test for tannins in the sap. In literature, tannins are known to increase formation of capillary vessels. So, not only does the tannins phytochemical have the property of wound healing, but also repair damages brought about by bacterial infections.
Furthermore, flavonoids which were confirmed to have been present, as well as the sap, the ability of stimulating reepithelialization, which is the ability for a wound to heal appropriately and quickly. So, having confirmed the presence of these phytochemicals means that the sap does have properties that both arrest the growth and spread of bacteria in wounds, but also possess the capacity to regenerate skin tissues. stimulate the formation capillary vessels and allow the wound to heal quickly and appropriately.
In terms of the feasibility of the project, we were aiming at developing a sugarcane banana sap antiseptic, since currently in the Philippines there is the development of ethanol that is derived from sugarcane. And in literature, ethanol has been used to precipitate out antimicrobial phytochemicals in plants. And bananas have been found to contain these phytochemicals. If we can continue this project further, we would like to see a novel and synergistic approach to wound treatment by combining these two bioresources found in the Philippines.
This way, we can combine both the economic aspect of our project by producing much cheaper alternatives to commercial antiseptics for rural communities, as well as understand more deeply how we can make use of the chemistry of these plants and utilize them as an important bioresource.